everyone, welcome to Backbenchers. I hope everything is going well with you all and I hope you all are fine. Today we are going to be discussing about the third portion of the chapter, Absorption by Roots. We have already discussed about the first two portions of this chapter. So if you haven't checked the videos, then make sure you go and check them out because they are already been sent. Starting with today's session, let us quickly have a look into the division of the chapter. This was the first portion which we have already finished and the second portion which we have finished as well. Today we are going to be discussing about this portion, that is osmosis and its types. And then these are the rest of the divisions. After finishing all of the divisions, I have kept a separate doubt class for all of you so that I can answer the doubts which you have asked in the comment section. So whatever doubts you're having, you keep writing them in the comment section, and at the end of this video, at the end of this chapter, I'll be shortlisting the doubts which are most frequently and most important ones, and then I'll answer them in that particular video. Let's start with today's lesson, that is osmosis and its types. Here are some questions from the previous session. The first question is, which are the actual absorbing parts of the plant? Roots or root hairs? Both of this topic is already discussed in the previous session. This is the second question. State one point of difference between active transport and diffusion. In the previous video, more than one point of difference was discussed. So make sure you choose the most important one and then you write it down in your answer. And then this is the last question from the previous session, which is state the direction of active transport. This question is a bit tricky. I'll put a star mark on this question. Go through this question very carefully. Think about the answer because this question is not hard. This question is just tricky. And you all can answer this question, I know, if you have understood the portion of active transport well. So make sure you understand active transport first, and then this question is not that hard to be answered. So think about this question well. Let us start with today's session. We have osmosis today. Osmosis is the fourth process through which roots absorb substances from the ground to complete the process of absorption. So to understand osmosis, let us look into a particular diagram. This is a very simple diagram that I have chosen. First of all, let us see that what do we see in this diagram. First, we see a plant cell. So here is a plant cell because it has a semi-permeable membrane as a cell wall and a cell wall as well. So this is a cell wall. And then the semi-permeable membrane is already marked. This is a semi-permeable membrane. So we have a plant cell here. And then this is the surrounding of the plant cell. The area surrounding the plant cell. Now, let us understand the concentration of the substances in the plant cell and the surrounding. Inside the plant cell, higher concentration of salt molecules are visible. You can see very well in the diagram that there are more number of salt molecules inside the plant cell than there are outside. And in the surrounding, there are more number of water molecules than are inside the plant cell. So, in the cell, there is more solute. Solute is what? The salt molecules. And less solvent. Solvent is the water molecules. And if we look into the surroundings, it has the exact opposite configurations. It has more solvent and less solute. So now we have noted the concentration of the plant cell and the surroundings. But we only have to consider the concentration of solvent here. That is, let me just change the ink. We are only going to be focusing on the solvent here, that is the water molecules. So let us just look into the water molecules here. In the surrounding, we can see there's a huge number of water molecules present, but inside the cell, the number of water molecules is less than that of the surrounding. This is known as change in difference, or you can say difference in concentration between two things. And due to this difference in the concentration of solvent molecules, here we have taken the example of water, but actually it is solvent molecules. Any type of liquid it can be, leads to the process of osmosis. So what is osmosis? It is the process in which transfer of molecules. What type of molecules? Water molecules or solvent molecules. Transfer of molecules or solvent molecules, you can say. To be precisely, you have to mention solvent molecules in the definition when you will be writing it in your exam. So osmosis is a process in which transfer of water molecules takes place from a region of higher concentration to a region of lower concentration across a semi-permeable membrane. Higher concentration, which is the higher concentration here? Surrounding is the higher concentration. How surrounding is the higher concentration? Because we are considering here the water molecules. If we look into the surrounding has higher water molecules, that is higher concentration of water molecules than the plant cell. So plant cell has lower concentration. So if you remember the concept of heat, energy always flows from a higher body to a lower body. Exact same thing is happening here. Movement of water molecules is taking place. Water molecules are flowing from a region of higher concentration to a region of lower concentration across a semi-permeable membrane. This process is the exact opposite of active transport. If you remember the definition of active transport, it is the exact opposite, but it has a difference, a very vivid difference. And what is the difference? That is, osmosis only involves solvent molecules. That is, liquid molecules. Active transport involves movement of all types of substances, but osmosis does not allow that. Osmosis only allows the movement of liquid molecules through the semi-permeable membrane. So you remember this point because this is a very important point when you will be writing the difference between two processes like this. A very important point regarding osmosis is it only involves the movement of solvent molecules, that is liquid molecules. There is no action of solute molecules here. It only involves the solvent. Now we have already figured out the definition of osmosis. We know what is happening in osmosis, but still let us look into the definition once. Definition says that osmosis is the movement of only liquid molecules. Only liquid molecules. Let me just highlight that particular thing for you all. Only liquid molecules from their region of higher concentration to their region of lower concentration across a semi-permeable membrane. The presence of semi-permeable membrane is also very, very important. By that, we have understood that what is osmosis, and a very simple definition of osmosis also we have known. It is nothing out of the box. It is not difficult. You all know osmosis. You all have studied osmosis since class 8. So it should be very easy for you to understand. But this particular point is a very important point. Let me just underline that again so that you don't forget it. Only liquid molecules. This is a very, very important point. And apart from that, rest of the things are very same. Higher concentration to their lower concentration and then across a semi-permeable membrane. So a question might be asked to you all that if osmosis is the exact opposite of active transport, then why cannot we call it as passive transport? We cannot call osmosis as passive transport. Why? Because osmosis involves only the movement of water molecules. Active transport was a process which involved the movement of all types of substances. But osmosis strictly allows the movement of only water molecules. Only water molecules. Now, how long does osmosis continue? It continues till both the substances reach the equilibrium in the concentration of water or liquid molecules. It's the same like the flow of heat energy. If two bodies having different heat energy are kept in contact, then uh, heat flow is observed. The flow of heat continues until and unless both the bodies reach same temperature. So, let me just clear this area for you all so that I can explain you this point. Okay. It continues till both the bodies reach the same concentration of water or liquid molecules. So we will understand this with the help of the concept of heat. Very simple concept, you all know. Or let's not consider any concept. Let's just understand it on its own. Suppose here are two bodies. This is body number 1 and this is body number 2. And we have established a contact between the two bodies. Then transfer of substances are going to take place between the two bodies until and unless both of the bodies have equal number of products. So until and unless both the bodies reach equilibrium. Equilibrium state means having same number of substances or having the same concentration of water molecules, because we are talking about osmosis here. So same number of having same number of water molecules. Till that time, osmosis continues. It's a very common sense topic. You all can understand that it is going to continue until and unless both of the regions have equal number of water molecules. It's a very common thing to understand. So that was all about osmosis. Osmosis is a process in which transfer or movement of only liquid molecules takes place from the region of higher concentration to the region of lower concentration across a semi-permeable membrane. And the semi-permeable membrane is the plant cell membrane here. That was all about osmosis. Now, we are going to look into the types of osmosis. Osmosis are basically of two types. First is endosmosis. Second is exosmosis. Endosmosis and exosmosis. Now, whenever you will get the word endo in biology, you will have to remember one thing, that endo means inside. And whenever you're going to get the word exo in biology, then it is going to mean outside. If you remember the meaning of these two terms, then it will be very easy for you to just answer or understand the phenomenon that is going to follow. So endo means inside and exo means outside. Now by hearing the meaning of these two terms, you can understand that these two processes have something to do with inside and outside. So that we are going to be understanding. Let us look into this particular diagram. It's a very beautiful diagram which I found online. So it will be helping you a lot to understand these two processes carefully. First, let us look into this diagram. This is the first diagram. So we are going to look into this diagram first. This one, where I'm pointing this diagram. This is the first diagram, and we're going to look into this diagram. First, we can understand this is a plant cell. This, sorry about that. Let me just erase it. Okay. This is a cell wall, and this is a cell membrane. Cell membrane is semi-permeable in nature, you all know, and cell wall is free permeable in nature. And this is a vacuole. We have already discussed that plant cells have very large vacuole, which is filled with sap, cell sap. So in this diagram, what is happening? Water molecules are entering. This arrow indicates the entry of water molecules. So water molecules, entry of, let me just write it down, entry of water molecules. Water molecules from the surroundings are entering into the cell. And due to that, what is happening? The cell is becoming tight. That is, the cell is filling up with water more. Hence, the cell is becoming compact and tight. This is known as endosmosis. Why endosmosis? Because water is going inside the cell. Anything is going inside is known as endo. And the process is endosmosis. Why endosmosis? Because it is involving water molecules. Because it is involving the water molecules, the entry of water molecules. So when water molecules from the solution or from the surrounding, you can say from the solution or surrounding enter into the cell through the semi-permeable membrane, it is known as endosmosis, which is happening exactly in this case. Very simple to understand. Just from the surrounding, water molecules are entering into the cell, and that type of osmosis is referred to as endosmosis. It's not difficult to understand at all. Example of endosmosis in day-to-day life is when dry raisins are placed in water. If you have looked and seen in houses, dry raisins are placed in water before cooking, then what happens after some time, they swell up, and that is uh, endosmosis. They swell up because they have performed endosmosis. That is, water molecules from the outside, from the surrounding, have entered into their cell. That is very simply endosmosis. Similarly, exosmosis, the exact opposite of endosmosis is exosmosis. This is the water movement of water molecules outside the cell back into the surrounding. So when water molecules, these arrows indicate the exit of water molecules. Exit of water molecules. The exit of water molecules from the cell back into its surrounding is known as exosmosis. So when water molecules come out of the solution, come out of the cell into the solution through the semi-permeable membrane, it is known as exosmosis. From the into the solution, or you can say surrounding as well. So surrounding or solution, then that particular process is referred to as exosmosis. For examples, from real life examples we have taken here, this is fresh grapes placed in water. When fresh grapes are placed in water, what happens is the grapes shrink. Why? Because the grapes already have enough amount of water in them. If they are again placed in water, then further water comes out from them and the cell contents shrink. You can see in this cell that the cells are shrinking inside. in this direction. The cell contents are shrinking. Now, when the cell shrinks, that state or that phase is referred to as a different term, which we will be discussing in the later parts of the chapter. But here the cell is shrinking, and here the cell is expanding outside. That is, the cell is stretching. So it involves cell stretching, and this involves cell shrinking. Very simple definitions of endosmosis and exosmosis. When water molecules from the solution enter into the cell through the semi-permeable membrane, it is known as endosmosis, which involves the stretching of the cell or the bulging of the cell. You can say stretching or bulging. And when water molecules come out of the cell into the solution through the semi-permeable membrane, it is known as exosmosis, which involves shrinking of the cell shrinking. That was all about the types of osmosis. Apart from these, now we have few experiments to discuss about osmosis. There are basically two experiments to be discussed about osmosis. The first one is to demonstrate osmosis using thistle funnel experiment. Thistle funnel experiment is given in the book which you all are following currently. And the next experiment is to demonstrate osmosis using potato osmoscope experiment. This experiment is not given in your book, but potato osmoscope experiment is a very important experiment in class 10 because a lot of practicals are done based on this particular experiment. So it's better to know this experiment beforehand. And it's a very simple and easy experiment. You all can perform it at your home as well. So it will be fun to understand. So let us just look into the first experiment, which is by using thistle funnel. We are going to be using sucrose solution to demonstrate the process of osmosis in this experiment. What is sucrose solution? I'll be telling that definitely. Sucrose solution means sugar solution. So if you just see the term sucrose anywhere, then don't get scared. It just means sugar solution, right? So three apparatus are shown here. Three conditions are shown here. First, let us look into the figure A. Here we have taken a round bottom flask. This thing is called a round bottom flask. Let me just write it down for you all. Then this is a thistle funnel. This is a mouth stopper, or you can say stand. So let me just write stand here to hold the round bottom flask. This is a beaker. This is a beaker. The beaker consists of water, and the mouth of the round bottom flask, or the bottom of the round bottom flask, is covered with semi-permeable membrane, that is sealed with semi-permeable membrane. Inside the round bottom flask we have taken sugar solution. This sugar solution is 20% sucrose solution, that is, it has 20% of the solute in the solution. And then this is the initial level of solution. Then here's another apparatus. This apparatus is the same result of this A apparatus. Here we are going to observe that the level of solution has increased. So previously the level was this. It has increased this much. And now the final level of solution is this. And apart from this two apparatus, we have also taken a separate apparatus where we have taken water in both the places. This also consists of water, and this also consists of water. So both the places have water. Now this is the experimental setup. What is the experimental setup? A and B together make up the experimental setup, and C alone is the control setup. C is the control setup. What is happening in the experimental setup? First of all, we have taken a round bottom flask, a beaker, 20% sucrose solution, thistle funnel, semi-permeable membrane. These are the materials of the experimental setup. What we have done in the experimental setup, we have taken 20% sucrose solution inside the round bottom flask, and we have taken water in the beaker. Then we have just simply placed the round bottom flask in the beaker full of water. Then we have to let it sit for some time. As always, experiments involve. So this apparatus B is nothing but the result that we are going to obtain after letting the apparatus A to sit for some time. So apparatus A is the same as apparatus B, but after few time. After letting it sit for some time, when we are observing the apparatus A, we are referring it as apparatus B. So do not get confused that there are three apparatus. There are all total two apparatus. First is the experimental setup and second is the control setup. The experimental setup is shown in two ways. First is the initial step and second is the final step. So in the final step, we are observing that the level of solution inside the round bottom flask has increased little bit. That is all we are going to observe. And we are also going to observe one thing which is not given in this particular diagram, that is the level of water will fall in the beaker. When the apparatus A will be allowed to sit for some time, then we will observe that the level of water in the beaker has fallen and the level of water in the thistle funnel has risen. Let us look into the control setup. To check the difference between both the setups, we have taken a control setup. In the control setup, we have not taken any sucrose solution. We have taken water, both inside the round bottom flask and in the beaker as well. Now, what we can conclude from this change in the apparatus B of this experiment, we can understand very well that osmosis has taken place. How and why? How? That we can understand that water molecules have passed into the round bottom flask, crossing the semi-permeable membrane. So the level of water has risen here and the level of water has fallen in the beaker. Why? Because water molecules have gone inside the round bottom flask. Hence, there is a change in the rise or change in the level of water in both the apparatuses. Now, let us look into the observations carefully. In the experimental setup, that is in the A setup, we can see that the level of water will rise in the round bottom flask of apparatus B and the level of water in the beaker will fall. We have just discussed about this point. Second is the control setup. In the control setup, there is no change because we have taken water. We have taken the same liquid. Hence, there is no change in the concentration. There is no difference in the concentration of the two things. That is why no change is happening in the control setup. So what is the inference that we can draw from this particular experiment? The experimental setup has gone through osmosis. Why? Due to the difference in the concentration of liquids present in the round bottom flask, 20% sucrose solution, and in the beaker water. So in the experimental setup, there was a difference in the concentration between the two liquids, right? In one part, we have taken 20% sucrose solution, and in the other part, we have taken water. So there is clearly a concentration difference between the two liquids. Therefore, osmosis has taken place, and since the cellophane is semi-permeable in nature, therefore it has allowed the entry of water molecules. If you remember the previous picture, if you remember the previous slide, Then the semi-permeable membrane was here, the cellophane. We have taken cellophane as the semi-permeable membrane in this experiment. So you can add that name in the materials required. Now, let us move on to what we were reading. Since cellophane is semi-permeable in nature, therefore it has allowed the entry of water molecules inside the round bottom flask, but not the entry of sugar molecules into the beaker. Semi-permeable membrane, it only allows the entry of water molecules through it, and it does not allow the entry of sugar molecules. Why? Because sugar molecules are bigger molecules than water molecules. Therefore, it only allows water molecules. Hence, the level of water has fallen in the beaker and the level of solution has risen in the thistle funnel. That is why it is happening due to the presence of the semi-permeable membrane. And on the other hand, in the control setup, there is no change in the level of water. Why? Because we have taken, because of the presence of water molecules throughout. That is why there is no change in the level of both the liquids. So that is about the first experiment. Very simple experiment, very easy to understand. Let us look into the second experiment now. Acha, before going into the second experiment, we have two more things to know. We are considering a rubber sheet in place of cellophane paper now. We are considering and we are using a rubber sheet as the semi-permeable membrane in case of cellophane paper, and then we are performing the experiment again, performing the same thistle funnel experiment again to see and check whether osmosis takes place or not. What we are going to see is that the water in the round bottom flask and the beaker respectively remain same even after some time. That is, there is no change in the level of water. Even if we take two different solutions, that is sugar solution and water, there is no change in any level of any liquid. Why? Because rubber sheet is not a semi-permeable membrane. Rubber sheet is not a semi-permeable membrane. If it would have been a semi-permeable membrane, then water molecules would have crossed it and would have gone inside the thistle funnel. That means the rubber sheet has not allowed the entry of water molecules through it. Second, we are using muslin cloth in place of cellophane paper. Muslin cloth, we are trying to use muslin cloth as a semi-permeable membrane in place of cellophane paper. What we will observe that the water and sucrose solution get mixed together. The muslin cloth has large pores, therefore it allowed the free movement of both water and solution molecules to pass through it. Now muslin cloth is not acting as a semi-permeable membrane either, is definitely not a semi-permeable membrane. It is a freely permeable membrane. It is a freely permeable membrane with very big pores, which allows the entry of both water molecules and sugar molecules to pass. Hence, what is happening? We are observing that both the solutions are getting mixed together and then they're making a common solution in the whole apparatus. That is why we cannot use both of these things. That is why, therefore, rubber sheath and muslin cloth can't be used in the thistle funnel experiment because they do not act as semi-permeable membranes. Thistle funnel experiments. That was all about the first experiment. Next, let us discuss quickly the second experiment. Second experiment is a very simple experiment. We are going to demonstrate osmosis using a potato osmoscope. Osmoscope, what is an osmoscope? Osmoscope refers to an apparatus that demonstrates osmosis. So any apparatus that demonstrates osmosis gets the name of osmoscope. Let us look into the procedure. What is written in the procedure? Okay, let's not read the procedure at all. Let us quickly jump into the ingredients that we need or the materials that we need. We have a petri dish here. We have taken a petri dish. This is a petri dish. If you all do not know about it, then know that this is a petri dish. Let me just write it down again. And this petri dish is half filled with water. Very clearly visible, this petri dish is half filled with water. Now already we have our first material here. What we need is a potato. We need a potato to demonstrate this experiment. So we are going to take a potato from this home. You all can perform this experiment right now, right here with me. If you all are watching this video, please do it. It's a very fun experiment and it's very easy to do at home. And what we are going to do with the potato, we are going to cut the sides of the potato to give it a square shape. So we are going to cut the sides of the potato and give it a square shape, and then we are going to place it, and then no, before placing it in the petri dish, we have to scoop a cavity out as well. So we have to scoop a cavity out like this, and the potato is going to look like this. So this is cavity that we have scooped out, and we have given the potato as square box-like shape. In this cavity, we have to put sugar solution. Suppose we have put sugar solution up till this level. We have put sugar solution till this level. Now, we have to mark the level of this particular sugar solution using a pin. So here we are using a pin. This thing is a pin to mark the level of sugar solution. Now we have to place this whole thing in the petri dish. So that is what we are going to do now. We are simply going to place the potato osmoscope in the petri dish. But you have to remember one thing, that the potato should dip the water completely inside the petri dish. So this is the potato. Then this is the scooped in cavity that we just now scooped out using a knife. And then we have the square shaped potato like this. This is still a potato. We have to peel it definitely. First we have to peel the potato and then we have to give it this particular shape. We have scooped a cavity out. Now what we are going to do is we are going to pour sucrose solution in this cavity up to a low limit, and then we are going to mark the level of the solution using a pin. Suppose this is a pin. Then what we have to do? Just wait and let the apparatus sit undisturbed for some time. After some time, what we are going to observe? After some time, a very fun thing is going to be observed by all of us in the potato osmoscope. What is that thing? We are going to observe that the level of sucrose solution or the level of solution inside the cavity has risen to this new level. So whatever the new level is, we are going to mark it with another pin to clearly check the difference between the two levels of solutions. This is level 1 and this is level 2. So there's a change in the level of solution, and basically, to be precise, there is a rise in the level of solution. Why is this rise happening? Because of osmosis. Very simply understood. How is osmosis taking place and why? This is a potato. Potato is a part of plant cell. Plant cells have semi-permeable membrane in them. So potato here is acting as a semi-permeable membrane. The cells of the potato here are acting as semi-permeable membrane, and they're allowing the movement of water molecules from the petri dish inside the potato. Then they're going and mixing with the sugar solution, and therefore the level of sugar solution is increasing. So that is what is happening here. Now let us read the procedure what is written. First we have to cut the sides of potato to give it a square shape, but before cutting the sides of the potato we have to peel the potato as well. So make sure we first peel the potato and then we give it a square shape, very simply. Then we have to scoop out a cavity from the potato so that it looks like the image at the right, basically this thing, so that it looks like this image, like this. Then we have to pour the solution. What solution we have taken here? We can take 20% sucrose solution, 20% sugar solution, or we can just simply take sugar solution. But if you all want to carry out this experiment at your home, then you do not need 20% sucrose solution. What you can simply do is you can cut a potato, peel it, and then give it a square shape, make a hollow cavity in it, and then use powdered sugar. Put powdered sugar in the cavity, mark the level of the sugar with a pin, then place it in a you can say plate or bowl which is half filled with water. You have to dip the potato there, and then you have to let it sit for some time, and then you can clearly notice the observation, clearly notice the changes. The water is going to rise. If you do not have powdered sugar, you can just use cubed sugar as well. But powdered sugar is preferred when you are performing this experiment at home, because powdered sugar does its job more quickly. Because it is already in the powder form, it is more easily diffused, it is more easily melted. So the process happens quickly. Rest, you can simply use a sugar cubes or anything at your home. Sugar cubes basically you have to use. You can use salt as well. So if you use this at home, you can perform this experiment and clearly see osmosis taking place. So if you're performing this experiment at home, then you can use sugar solution, or basically you can use cubes of sugar. Now that was the procedure. Let us move on to the inference and observation. So we have already discussed what are we going to observe. We are going to observe that the level of water, level of solution inside the cavity of the potato osmoscope has risen. What is the inference that we can draw? Osmosis has taken place, definitely. We already discussed just now. The cells of potato control cell membrane. Very important explanation. You have to remember this portion because this is a very simple explanation. It will help you to understand. As it is a plant cell which is a semi-permeable membrane, hence the entry of water from the petri dish to the cavity. Therefore, water is entering from the petri dish to the cavity of the potato osmoscope. Here cavity of, sorry, here cavity of potato osmoscope we are taking. That was all about the experiment. Now is a small, very small definition. You have to just remember it. I have already said you when I was talking about potato osmoscope. Osmoscope is basically an experiment which demonstrates osmosis. It can be any experiment. I'm sorry about that again. Osmoscope is any apparatus that demonstrates the process of osmosis. In the previous experiment, osmoscope was potato. Since we were using potato to demonstrate osmosis, it was known as potato osmoscope. That was all about today's session. It was a very fun session. We had a lot of fun discussions about experiments. I hope you've understood the session well. Make sure you put your doubts in the comment section. And if you have found out this video useful, then definitely share with all of your friends. Share in a very high number and like this video and subscribe to my channel for getting more such videos. Till then, stay safe, stay happy, take care of yourselves. Peace.